Yay, last 3 lectures. So happy. Anyway, it's hard work. Okay, so let's start with muscarinic receptor. Let's say it's on the heart and it has inhibitory effect. Which channel do you think it's on this muscarinic receptor? So if it's inhibitory, it's always potassium. And if it's excitatory, it's sodium. And so let's start with, I'll start with this table later, but just give you an example. In somatic nervous system, we have the skeletal muscle, and in skeletal muscle, it's the ionotropic receptor. And what's the difference between ionotropic receptor and metabotropic receptor? So I'll give you a few seconds. So the difference is that the ionotropic receptor, the something binds to it, it's the acetylcholine bind to the receptor, and the channel is the receptor. It doesn't require a GDP to move to another enzyme and then signal the channel on the membrane to open up. So for ionotropic, the receptor is the channel. And metabotropic is when something binds to the receptor and it has to have something to move and signal the channel on the membrane. The membrane and the receptor is not the same protein. And on skeletal muscle, we know that it's always excitatory. And like I said earlier, if it's excitatory, it's always sodium. So it's sodium channel on the skeletal muscle. Then let me ask you a question. So if I only tell you an unknown receptor is ionotropic, are you going to know that it's either excitatory or inhibitory? It's a question to ask you. If I'm only telling you this channel is ionotropic, am I telling you this channel is excitatory or inhibitory? The answer is no. If ionotropic, the definition is only telling you the channel and the receptor is the same protein. It doesn't tell you whether it's excitatory or inhibitory. That's a common mistake people make. And before going into this table, let me just categorize the neurotransmitter for you and the receptor. So we are talking about two groups of neurotransmitter and receptor. One is cholinergic. The other one is adrenergic. In the first group, cholinergic, they all have different neurotransmitter and receptor, the same thing, just two different groups. And in cholinergic, the neuron is just called cholinergic neuron. And for neurotransmitter is acetylcholine. If we are talking about cholinergic neurotransmitter, we are talking about acetylcholine. And if we're talking about cholinergic receptor, which is where acetylcholine bind to, it's either nicotinic or muscarinic. That's the first group. Acetylcholine never binds to adrenergic receptor because they're in different group. Just separate them completely. And let's talk about the second group, adrenergic. The neuron is just adrenergic neuron. And adrenergic neuron release adrenergic neurotransmitter, which are epinephrine or norepinephrine. And they bind to only the adrenergic receptor because they're in the same group. And we have alpha and beta. So let me mention it again. Acetylcholine only bind to nicotinic receptor or muscarinic because they're in the same group. And epinephrine, norepinephrine only bind to alpha and or beta receptor because they're in the same group. And nicotinic is always excitatory. Muscarinic depends on the location. It has either excitatory or inhibitory. And for adrenergic receptor alpha or beta, it depends on the location. We do not know whether it's excitatory or inhibitory. Okay, back to this table. Let's start from the top. Somatic nervous system. So this one is the brain sends a neuron and then it creates acetylcholine, and acetylcholine bind to skeletal muscle to tell the muscle to contract. So what type of neuron is this? Since it's secreting acetylcholine, it belongs to the adrenergic group. So this neuron is adrenergic neuron. It releases acetylcholine, and what type of receptor is on the skeletal muscle? It's the nicotinic receptor, because we know that it's always excitatory on the skeletal muscle. When acetylcholine binds to nicotinic receptor on the skeletal muscle, the sodium channel opens up and sodium rush into the skeletal, rush into the neuron and then performs muscle contraction. Rush into the cell, I should say. Okay. And in autonomic nervous system, we start with sympathetic division. Let's look at the first line. This neuron is secreting acetylcholine. What type of neuron do you think this neuron is? It's adrenergic neuron because it's secreting acetylcholine. And what receptor is on the second order neuron? When I say second order neuron, is this one is the first order neuron and this one is the second order neuron. And between the ganglion, we have receptor that's receiving the acetylcholine. Do you think it's nicotinic or muscarinic? So the answer is nicotinic, because between neurons, we always want the communication to pass on. So we want it always to be excitatory. And then this neuron is releasing norepinephrine. What type of neuron do you think this is? So it's adrenergic neuron because it's releasing norepinephrine. And what type of receptor on the smooth muscle do you think will receive this norepinephrine? So it's adrenergic receptor. And adrenergic receptor, we have either alpha or beta. Let's look at the second line in sympathetic division. What's this neuron that's releasing acetylcholine? It's cholinergic neuron. And what type of receptor is on the adrenal medulla? So between these two, this adrenergic medulla is like the second order neuron, so it's nicotinic. And adrenal medulla release epinephrine and norepinephrine. It goes through the blood vessel. If this goes through the blood vessel and it's released by an organ, a gland, this one, which makes these chemicals hormone. And hormone binds to a gland, let's say. What type of receptor is on the gland to receive epinephrine and norepinephrine? It's adrenergic receptor. It's either alpha or beta. You don't need to differentiate which exactly is on the gland or smooth muscle. You just need to know it's the adrenergic receptor receiving the epinephrine or norepinephrine. Okay, last line, parasympathetic division. We have the first order neuron releasing acetylcholine. So this one is cholinergic neuron. And between the neuron, at the ganglion, what receptor is on the second order neuron? It's the nicotinic. We always want excitatory. We want the message to be passed on. So it's nicotinic here. And this second order neuron release acetylcholine, which makes this second order neuron also a cholinergic neuron. And this acetylcholine goes bind to the receptor on the cardiac muscle. What type of receptor is that? It's muscarinic. All right, something else to mention at the end. On the parasympathetic, so for all the autonomic nervous system in adrenergic group, on the smooth muscle gland, cardiac muscle, on these muscle, they all have both they all have both adrenergic and muscarinic receptor. They have both cholinergic receptor and adrenergic receptor. They have both receptor on the cell. All right. So let's go into something else. Glutamate, GABA. Yeah, we're going into details about glutamate. So glutamate is produced in the brain, and it's going to bind to ionotropic glutamate receptors. It's a neurotransmitter that's between the neurons in our brain. It's receptor ionotropic. Receptor and the channel is the same protein. It doesn't need to signal other protein to open up the channel. The channel is the receptor. And it has an excitatory effect. So you can guess it's sodium channel. And if we have a high level of glutamate and low level of GABA, GABA is the inhibitory, it's the opposite of glutamate. It has inhibitory effect. So we have too much glutamate and low level of GABA, that will lead to epilepsy. And glutamate is used during memorization. How we remember things is through repetition. Why repetition helps? Because by repeating something, our brain knows, okay, this thing is important. I'm going to enhance this pathway. How do they enhance it? Is through producing more glutamate at the synapse of the neuron and by inserting more receptor at the postsynaptic neuron. If we repeat something more, the enhancement is called long-term potentiation. And if we don't repeat it and over time this pathway is slowly, we say depressed, it's the process of we produce less glutamate and some receptors are removed. So that's called long-term depression. In the test, if you are asked to explain what's long-term depression, you need to explain how the number of the glutamate at the presynaptic neuron and the postsynaptic neuron has less receptor. Glutamate decrease and less receptor at the postsynaptic neuron. Don't explain it as an illness. Okay, so this whole process is called synaptic plasticity. And this graph shows if we have this little amount of glutamate, then the neuron won't have action potential, won't fire, transfer the information to the next neuron. And when we have more glutamate, the pathway is made easier to transduct the information. Alzheimer's is related with glutamate. When we have too much glutamate at the synapse, that kills the postsynaptic neuron. How this happens is because there is no reuptake of glutamate in Alzheimer's disease patients. And this killing the postsynaptic neuron is called excitotoxicity. GABA. GABA has also ionotropic receptor, and it has an inhibitory effect. The channel it's using is chloride. If it's chloride, and it has an inhibitory effect. Do you think it's depolarizing the neuron or repolarizing the neuron? The answer is depolarize, because look at this graph. When we fires, oh, sorry, it's repolarize. I made a mistake. Repolarize. So when we want to excite something, we depolarize. And this chloride will keep adding because if we open up the channel, let the chloride in, it's something negative. We're making this graph going down more. So that's repolarization, makes the action potential harder to reach. We have two medications that will mimic the effect of GABA. In normal times, GABA will bind to the chloride channel. And if we are using barbiturate, for example, both of the medication works the same. So they bind to a receptor on the this channel receptor, and then that will make the benzyl bind to chloride channel easier. And the difference between this medication is that barbiturate open the channel for longer. It just keeps the channel open, and chloride keeps rushing in. And for benzyl, it just make the channel like this, open up frequently but short. So the chloride rushing a little bit, and then wait for a bit and then comes in. So that makes benzo more stable. The patient doesn't have a drastic change when taking benzo, because too much inhibitory will decrease breathing rate and heart rate. And increasing GABA shuts off a part of the brain that function for anger, fear, aggressive behavior, and sex. That's just a side note, not too important. All right, let's talk about Parkinson. This is a pathway that's important to understand the medication, but you do not need to know exact these terms. It's just to know an idea that, okay, L-DOPA is coming, all these dopamine, norepinephrine, epinephrine came from this original form. Depends on the location, a gland will decide for example, the adrenal gland will decide to make dopamine to norepinephrine or later make epinephrine, depend on the use. And in Parkinson, we give the Parkinson patient L-DOPA. We do not give dopamine directly because dopamine is too big to cross the blood-brain barrier. And we need to put more dopamine in the brain. That's why we give something before it becomes dopamine. We give L-DOPA. It's able to cross the blood-brain barrier and become dopamine in the brain. And beside that, we give carbidopa. The reason we give carbidopa is that we want this L-DOPA, when we give to the patient, some will go into the peripheral nervous system and become dopamine. And that will lead to GI upset. We do not want that. That's why we give carbidopa. Carbidopa cannot cross the blood-brain barrier. It will just inhibit the L-DOPA in peripheral nervous system to become dopamine. And that makes more L-DOPA move to the blood-brain barrier and leads to more dopamine in the brain. Alertness. So epinephrine and melatonin will go to act on pons and then act on reticular formation to have alertness. I don't think that's too important. And this adrenergic receptor I already mentioned before I explained the table. This one, I'll just give you an example. We all know the medication beta blocker, it slows down the heart rate. If we are blocking the beta receptor, and it will decrease the heart rate, and that makes this beta receptor excitatory. Slow it down and rethink about it. We're blocking a receptor, and the result is decreasing heart rate. If we do not block this receptor, which makes this receptor having the function of increasing the heart rate. So on the heart, we have beta-1 receptor. That has the effect of increasing heart rate. It's just an example for you to understand alpha and beta, and you do not need to know which one works as which. Okay. So adrenal gland has only adrenergic receptor. I'm having, because earlier in the table, it's saying this acetylcholine is acting on the adrenal medulla. But acetylcholine will never bind to adrenergic receptor. So that's weird. So I'm still skeptical about this, but it's not too much too important for the exam. I will double check with the teacher. Let's just move on for now. And dopamine. Dopamine nucleus accumbens act on the nucleus accumbens. It gives pleasure, reward, motion. Oh, never mind. The feeling of reward. And because in Parkinson patients, there's degeneration of this part of the brain that makes the level of dopamine lower. That's why they cannot perform smooth motion. And there is increase. So actually the number of the acetylcholine did not change. It's just something else decrease makes the acetylcholine high compared to dopamine. This one I mentioned before. And we use MAO-B is a medication. MAO is an enzyme that will break down the dopamine at the axon terminal between neurons. The gap is called axon terminal. And by using this medication, we remove this enzyme. That will keep this dopamine longer at the axon terminal. That's what we want for Parkinson patient, so that the receptor at the postsynaptic neuron has more time to receive that dopamine. Another way to help the Parkinson patient is to decrease the cholinergic, the acetylcholine. How? It's by using the anticholinergic medication. Or we use this acetylcholinease to break down the acetylcholine in, no, never mind. No, let me explain this again. So let me explain the how acetylcholine works. So first, at the synapse, you have acetylcholine, and the enzyme helps to break choline and acetate. Acetate goes out into the system, and then choline is recycled into the presynaptic neuron. And we also get choline from the diet. And the acetate came from the respiration, yeah. And both of them combine, and then they make acetylcholine. Acetylcholine with the income of calcium, and that neurotransmitter is released into the synapse. It binds to the acetylcholine receptor, and then the sodium channel open up. Then this cycle repeats. One of the medication is to block this enzyme so that the acetylcholine won't break down. It will just keep its place here at the synapse. No, but we're trying to decrease the number of acetylcholine. Let me double check the medication. This is lecture 10. Hello. Sorry, no. Who are you looking for?
Can you hear who?
Yeah, that's me.
Okay, I'm calling from Dr. Klein's office.
Yes, hi.
Yes, hi. You sent us a message that you're going to be away on to cancel your pop test appointment.
Yes, I'll be away.
Okay, very good. Cancel it. Just when you come back, to just call the clinic. We don't have the schedule just yet, but just when you get back to call us, and we'll put you on the appointment.
Okay. The number is on the Google map. Okay. Just one sec. Yeah, you can tell me.
Okay, it's 514
514
312
312
0333.
514 312 0333.
Yes.
Okay, thank you.
Okay, thank you so much.
Bye. I'm checking what's the receptor here. It didn't say. Okay. I think I saw it in other. Maybe it's not for Parkinson. Soon after recent. Anyways, the process I mentioned was correct. It's just I'm not sure which medication will affect this cycle. Depending on the exam, which medication, usually the meaning is in the name. I think it has something to do with blocking this enzyme or, I don't know, prevent the reuptake. Let's go back. So here at the same cycle, if we increase the calcium concentration here, and that will lead to closing down of the voltage-gated sodium channel at the beginning of the neuron. That will make this neuron harder to reach action potential, and the message won't be passed on. That will also lead to muscle weakness. Nucleus accompanying brain is the award center, is where the dopamine bind to. All right, let's move on to serotonin. Serotonin is a happy hormone that's secreted in GI system, and it travels through bloodstream to the brain. And we have this medication, SSRI. It prevents the reuptake of serotonin. So more serotonin will be left at the cleft, at the axon terminal, which makes the person happier. And lastly, we have endorphin endogenous opioid. It's a natural hormone that's produced by our body. So the story goes with we step on a nail, and this blue line is the normal pathway where you will feel pain. So this nociceptor is stimulated and it transferred to, this is first neuron communicate with second order neuron and up to the brain. The brain will make you feel pain. However, if the pain is so traumatic, the brain is trying to protect you. When it reaches the brain, it will send down a descending neuron, and this descending neuron will secrete some chemicals here that will hyperpolarize the second order neuron, makes this neuron, if the graph goes like this, it's a little hum to reach the action potential. Hyperpolarization makes it more negative. So it's harder to have action potential. So the passage, the message is cut in between. And at this end, there will be less neurotransmitter being produced. So both way, it makes this communication harder. That's endorphin endogenous opioid that's released here at the green. That's it for this lecture. 3 muscles. What are the 3 muscles we have? Cardiac muscle, smooth muscle, and skeletal muscle. Agonist and antagonist. It's when we bend our elbow, the bicep is the agonist. Agonist is the muscle that's contracting, and the tricep is the antagonist that's assisting. It's basically the opposite of agonist. Ligament. Ligament is bone to bone. Tendon is muscle to bone. And for muscle cell, they are multinuclei. Multinuclei. Muscle can only contract and relax. It's neurogenic. Has to be stimulated by nervous system. Just good to know. These are all the background information. And for this, you do not need to know the order of which one is the most outside, superficial and deep, not for the exam. Okay, so here the point of origin is always the one that closer to core, and the point of insertion is the one further away. This one you need to know for the exam. In our muscle system, when we talk about skeletal muscle, now we start with skeletal muscle. We have 3 motor units. Number 1 has more capillary, more mitochondria, is the smallest one. That usually is at work when we do aerobic exercise. That's more than past 15 minutes. We use this aerobic. It has lots of capillary. It's for perfusion, and mitochondria is for consistently providing energy. That's why if you do marathon, this type is more exercised. It looks like you can think of it as beef type muscle. And let's talk about the third one. So that's the 2 extreme type. So I'll mention the third motor unit first. Third motor unit is the one that's the biggest. It's strong, but it gets tired easily. It doesn't have much. It has less mitochondria. It has less capillaries. Doesn't provide much consistent energy. It has creatine inside, the phosphocreatine, and less capillaries. So it's a chicken type muscle. And the second motor unit is in between 2 of them. It's the intermediate. Usually it's used for daily living, like standing, sitting. And in one muscle, we have the parts myosin, it's this blue one, and actin is the green one. So they swipe the myosin. Actin swipe the myosin. Myosin swipe the actin to perform contraction. I will go into detail later. It's here. So in muscles, we have smooth ER too, like other cells, but it's called sarcoplasmic reticulum instead. Why we mention smooth ER here is because in muscle, they store calcium, and we need calcium to perform contraction. You will see why it's important. So sarcomere is the meaning is muscle segment. This one is one segment. And we have the myosin and the actin. I think so, yeah. And myosin will swipe the actin. And the closer they swipe the actin together, is contracting the muscle. So first we have the release of calcium from the sarcoplasmic reticulum, and then that will signal acetylcholine, allows acetylcholine. Remember, at the end of the neuron, we need calcium to release the neurotransmitter. And neurotransmitter is released acetylcholine, and it binds to, it goes into the neuron of the skeletal muscle, and then allows ATP to bind to the myosin. The blue circle is the ATP, and when it binds to the myosin, it gives it power to move actin, swipe the actin more towards where we want. We perform contraction, and this swiping movement is called cross-bridge action. And in heart, it's a bit different. So we have these lines. This line is covering the binding site that we want for contraction, muscle contraction. So we want, if we want to perform contraction in heart, we need somewhat of an action that removes this line. This line is called tropomyosin. And how do we remove this line? It's by binding the calcium to the point, the green point, troponin. It's a receptor. Calcium bind to troponin, and this orange line will move away. When it's moving away, we release the myosin binding site so that myosin is able to bind to this site and swipe the myosin more towards each other and perform contraction. Next. This one are review, so I don't need to mention it again. Calcium stored in sarcoplasmic. So in muscles, it's always in the smooth ER, and smooth ER in muscles are called psychoplasmic reticulum. Excitation. When acetylcholine is released by the presynaptic neuron, it goes into this T tubule, and then it will signal the sarcoplasmic reticulum to release calcium. And calcium will further help removing the orange line. That's called trypsinogen. Tropomyosin, and then by reviewing the myosin binding site, and myosin bind to this binding site and perform contraction. There's no calcium in smooth muscle in smooth ER of smooth muscle. We are moving on to smooth muscle later. Before that, let me just explain the energy system. So if you are just a quick, like 1 second movement, that doesn't require lots of energy. So we will use the existing ATP in the muscle, that's ATP. And then if we are just contracting a bit more up to 3 seconds, then we're using the second system called creatine phosphate in the muscle. That's anaerobic, the weight training. For 3 seconds, we use the existing creatine phosphate in the muscle to create ATP. And then creatine becomes the waste product. Then if it's more than 3 seconds, we move on to the next energy system, lactic acid system. That uses the carbohydrate, the glycogen in the liver. Glucose is in the muscle and in the bloodstream. And that will create lactic acid. That can hold you up to 3 minutes. And if it's more than 3 minutes, we need an aerobic system that will use carb, fat, protein, and carbon dioxide, water will be the byproduct. This one is just breaking more into details what happened in the creatine phosphate system. not too important, just go through it. Creatine phosphate become creatine kinase and produce creatine and ATP. So this ATP is used, but little amount of ATP. So we use creatine, the waste product of the creatine phosphate system, to measure EGFR because this one is more stable, and creatine urea is not stable because it will increase if we eat too much protein. Lactic acid. I already mentioned this, so I'll skip. We do not need to know the respiration, the cell respiration. I just put it here for you to have a general image of how aerobic energy system happen. This one is, so in the cardiac muscle, we have gap junction. That means the cell and cell are very close to each other and they use direct communication. When for example, the adrenergic receptor is stimulated, that will increase the heart rate. And that is sodium channel. So sodium channel opening up, and one cell doesn't require much time. It's instinct. It sends the sodium down to next muscle cell, and that with the help of gap junction. And if we know that at the beginning of the heart, it's SA node. The heart muscle is always autorhythmic. It doesn't need the brain to control it. So if everything else is broken, we have SA node and the heart will beat by itself, the heart muscle, around 90 to 100 BPM. And AV node, if everything else is not working, it will pump around 50 to 60 seconds. And this is the neuron graph for heart. The the orange line is when the funny sodium channel. This channel is specific on heart. Just get an idea. No need to memorize. So when the funny sodium channel open up on the heart, it helps it to depolarize. That's what this graph is about. And for this graph is the pink line is how much time the heart needs to rest when the sympathetic nervous system is triggered. You see it's very short, and as we go into parasympathetic, the resting time is longer. Between each contraction, one beat takes longer. Here's how it goes in the neuron of heart muscle. So sodium rush in and we have action potential. As we reach the top, we have sodium going out, but at the same time we have calcium going inside. That's why we get this plateau. It's when the calcium is rushing in and sodium is going out. It slows down for the heart to contract. And this is what happens in the heart muscle. Calcium, a little bit of calcium is moving in to the cell, and then it signals the sarcoplasmic reticulum, the smooth muscle, to release more calcium, and that will lead to like ATP attaching to the myosin, and myosin binds to the calcium binds to the tropomyosin, and then calcium binding to troponin, and tropomyosin is revealing the myosin binding site, and myosin bind to this binding site, moves the actin to perform contraction. In conclusion, that was the process of cross-bridge cycle, the moving part. And the calcium channel blocker will stop the process I just mentioned because it blocks this channel. Smooth muscle, hanging there, we're almost done. Smooth muscle looks different than other muscle. And we found smooth muscle in peristalsis, vasoconstriction, bronchoconstriction. So you see that the myofilament, some has myofilament and some doesn't. So when the smooth muscle wants to contract, it doesn't contract uniformly. It's like twisted. But it has the same parts as other muscle, actin, filament, actin, myofilament, it's the same idea. However, the smooth muscle, the sarcoplasmic, I forgot what's it called, reticulum, doesn't have much calcium, only 10%. The most calcium comes from the extracellular fluid. And the process is similar. Calcium comes in, the calcium comes out from the sarcoplasmic reticulum, release some calcium, and this calcium will activate calmodulin. Calmodulin is a special thing in smooth muscle that will eventually help with ATP attaching to the myosin and swipe. It's the cross-bridge cycle again, and contraction happens. I just put this image here if you want to know what happens in between the calmodulin and the ATP attaching to filament. It's not part of the exam, but if you want to get an idea, that's great. So calcium will activate calmodulin and then activate the kinase, and kinase help ATP attaching to the myosin. And in smooth muscle, unlike the other, is each cell has to be stimulated. It's unlike the heart muscle that has gap junction, or skeletal muscle that will contract. They have multinuclear. Their skeletal muscle cell is really long, but smooth muscle, single unit, each unit has to be stimulated to have contraction. Multi-unit smooth muscle have to stimulate every single cell. Yeah. That's it. Thank you for listening.